Unit 5, Human Physiology. We will be covering total number of 6 chapters, which are breathing and exchange of gases, body fluids and circulation, excretory products and their elimination, locomotion and movement, control and coordination, chemical coordination and integration. The reductionist approach to study of life forms resulted in increasing use of physicochemical concepts and techniques. Majority of these studies employed either surviving tissue models or straightaway self-resistance. An explosion of knowledge resulted in molecular biology. Molecular physiology became almost synonymous with the biochemistry and biophysics. However, it is now being increasingly realized that neither a purely organized approach nor a purely reductionist molecular approach would reveal the truth about biological processes or living phenomena. Systems biology makes us believe that all living phenomena are emergent properties due to the interaction among the components of the system under study. Regulatory network of molecules, supramolecular assemblies, cells, tissues, organisms, and indeed populations and communities each create emergent properties. In the chapters under this unit, major human physiological processes like exchange of gases, blood circulation, locomotion, and movement are described in cellular and molecular patterns. The last 2 chapters coordination and regulation of the body units at the organismic level. Alfonso Forti, Italian anatomist, was born in 1822. Forti began his scientific career studying the cardiovascular systems of reptiles. Later, he turned his attention to mammalian auditory systems. In 1851, he published a paper describing structure located on the basilar membrane of the cochlea, containing hair cells that convert sound vibrations into nerve impulses, the organ of Cortez. He died in the year 1888. Alfonso Cortez, 1822 to 1888. To our chapter 14, Breathing and Exchange of Gases. As you have read earlier, oxygen is utilized by the organisms indirectly to break down simple molecules like glucose, amino acids, fatty acids, etc., to derive energy to perform various activities. Carbon dioxide, CO2, which is harmful, is also released during the above catabolic reactions. It is therefore evident that O2 has to be continuously provided to the cells, and CO2 produced by the cells has to be released out. This process of exchange of O2 from the atmosphere with CO2 produced by the cells is called breathing, commonly known as respiration. Place your hands on your chest. You can feel the chest moving up and down. You know that it is due to breathing. How do we breathe? The respiratory organs and the mechanisms of breathing are described in the following sections of this chapter. 14.1 Respiratory organs. Mechanism of breathing vary among different groups of animals, depending mainly on their habitats and levels of organization. Lower invertebrates like sponges, ciliates, flatworms, etc. exchange O2 with CO2 by simple diffusion over the entire body surface. Earthworms use that moist cuticle and insects have a network of tubes, tracheal tubes, to transport atmospheric air within the body. Special vascularized structures called gills, brachial respiration, are used by most of the aquatic arthropods and mollusks, whereas vascularized bags called lungs, pulmonary respiration, are used by the terrestrial forms for the exchange of gases. Among vertebrates, fishes use gills, whereas amphibians, reptiles, birds, and mammals respire through lungs. Amphibians like frogs can also respire through the moist skin. Cutaneous respiration also. 14.1.1 Human Respiratory System. We have a pair of external nostrils opening out above the upper lips. It leads to a nasal chamber through the nasal passage. The nasal chamber opens into pharynx, a portion of which is the common passage for food and air. The pharynx opens through the larynx region into the trachea. Larynx is a cartilaginous box which helps in sound production and hence called the sound box. During swallowing, glottis can be covered by a thin elastic cartilaginous flap called epiglottis to prevent the entry of food into the larynx. Trachea is a straight tube extending up to the mid-thoracic cavity. It divides at the level of the fifth thoracic vertebra into a right and left pulmonary bronchi. Each bronchi undergoes repeated divisions to form the secondary and tertiary bronchi and bronchioles ending up in a very thin terminal bronchioles. The trachea, primary, secondary, and tertiary bronchi and initial bronchioles are supported by incomplete cartilaginous rings. Each terminal bronchiole gives rise to a number of very thin, irregular walled and vascularized bag-like structures called alveoli. The branching network of bronchi, bronchioles, and alveoli comprise the lungs. Figure 14.1. Sectional view of the left lung is also shown. Epiglottis, larynx, trachea, pleural membranes, alveoli, pleural fluid, bronchiole, diaphragm, lung, third end of the rib, and bronchus. We have two lungs which are covered by a double-layered pleura, with pleural fluid between them, which reduces friction on the lung surface. The outer pleural membrane is in close contact with the thoracic lining, whereas the inner pleural membrane is in contact with the lung surface. The part starting with the external nostrils up to the terminal bronchial constitute the conducting part, whereas the alveoli and the ducts form the respiratory or the exchange part of the respiratory system. The conducting part transports the atmospheric air to the alveoli, clears it from the foreign particles, humidifies, and also brings the air to body temperature. Exchange part is the site of actual diffusion of O2 and CO2 between the blood and atmospheric air. The lungs are situated in the thoracic chamber, which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by a vertebral column, ventrally by the sternum, laterally by the ribs, and on the lower side by the dome-shaped diaphragm. The anatomical setup of the lungs in thorax is such that any change in the volume of the thoracic cavity will be reflected in the lung pulmonary cavity. Such an arrangement is essential for breathing, as we cannot directly alter the pulmonary volume. Respiration involves the following steps. First, breathing or pulmonary ventilation, by which the atmospheric air is drawn in and CO2-rich alveolar air is released out. Diffusion of gases O2 and CO2 across the alveolar membrane. Transport of gases by the blood. Diffusion of O2 and CO2 between blood and tissues. Utilization of O2 by the cells of catabolic reactions and resultant release of CO2. Cellular respiration as dealt in chapter 12. Figure 14.1. Diagrammatic view of human respiratory system. 14.2 Mechanism of breathing. Breathing involves two stages: inspiration, during which atmospheric air is drawn in, and expiration, by which the alveolar air is released out. The movement of air into and out of lungs is carried out by creating a pressure gradient between the lungs and the atmosphere. Inspiration can occur if the pressure within the lungs, intrapulmonary pressure, is less than the atmospheric pressure. That is, there is a negative pressure in the lungs with respect to atmospheric pressure. Similarly, expiration takes place when the intrapulmonary pressure is less than the atmospheric pressure. The diaphragm and a specialized set of muscles, external and internal intercostals between the ribs, help in generation of such gradients. Inspiration is initiated by the contraction of the diaphragm, which increases the volume of thoracic chamber in the anteroposterior axis. The contraction of external intercostal muscles lifts up the ribs and the sternum, causing an increase in the volume of the thoracic chamber in the dorsoventral axis. The overall increase in the thoracic volume causes a similar increase in pulmonary volume. An increase in pulmonary volume decreases the intrapulmonary pressure to less than atmospheric pressure, which forces the air from outside to move into the lungs. That is inspiration. Figure 14.2a. Relaxation of the diaphragm and the intercostal muscles returns the diaphragm and sternum to their normal positions and reduce the thoracic volume and thereby the pulmonary volume. This leads to an increase in intrapulmonary pressure to slightly above the atmospheric pressure, causing the expulsion of air from the lungs. That is expiration. Figure 14.2b. We have the ability to increase the strength of inspiration and expiration with the help of additional muscles in the abdomen. On an average, a healthy human breathes 12 to 16 times per minute. The volume of air involved in breathing movements can be estimated by using a spirometer, which helps in clinical assessment of pulmonary functions. Figure 14.2 Mechanism of breathing showing inspiration, air entering the lungs, ribs and sternum raised, rib cage, diaphragm is contracted, volume of thorax increased. B. Expiration. Air expelled from lungs, ribs and sternum return to original position, diaphragm relax and arc upwards, volume of thorax decrease. 14.2.1 Respiratory volume and capacities. Tidal volume, volume of air inspired or expired during a normal respiration. It is approx 500 ml, that is, a healthy man can inspire or expire approximately 6,000 to 8,000 ml of air per minute. Inspiratory reserve volume (IRV), additional volume of air a person can inspire by a possible inspiration. This averages 2,500 ml to 3,000 ml. Expiratory reserve volume, additional volume of air a person can expire by forcible expiration. This averages 1000 ml to 1100 ml. Residual volume (RV), volume of air remaining in the lungs even after a forcible expiration. This averages 1100 ml to 1200 ml. By adding up a few respiratory volumes described above, one can derive various pulmonary capacities which can be used in clinical diagnosis. Inspiratory capacity (IC), total volume of air a person can inspire after normal expiration. This includes tidal volume and inspiratory reserve volume. Expiratory capacity, total volume of air a person can expire after a normal inspiration. This includes tidal volume and expiratory reserve volume. TV plus ERV. Functional residual capacity, volume of air that will remain in the lungs after a normal expiration. This includes ERV plus RV. Vital capacity, the maximum volume of air a person can breathe in after a forced expiration. This includes ERV, PV, and IRV of the maximum volume of air a person can breathe out after a forced inspiration. Total lung capacity, total volume of air accommodated in the lungs at the end of forced inspiration. This includes RV, ERV, PV, and IRV, or vital capacity plus residual volume. 14.3 Exchange of gases. Alveoli are the primary sites of exchange of gases. Exchange of gases also occurs between blood and tissues. O2 and CO2 are exchanged in these sites as equal diffusion, mainly based on pressure and concentration gradient. Solubility of gases as well as the thickness of the membrane involved in diffusion are also important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called partial pressure and is represented by PO2 for oxygen and PCO2 for carbon dioxide. Partial pressures of these two gases in the atmospheric air and the two sites of diffusion are given in Table 14.1 and Figure 14.3. Table 14.1. Partial pressures in millimeter Hg of oxygen and carbon dioxide at different parts involved in diffusion in comparison to those in atmosphere. Respiratory gas O2, CO2 in atmospheric air 159 0 in alveoli 104 40 blood deoxygenator 40 45 blood oxygenator 95 40 tissues 40 45. The data given in the table clearly indicates a concentration gradient for oxygen from alveolar to blood and blood to tissues. Similarly, a gradient is present for CO2 in the opposite direction, that is, from the tissues to blood and blood to alveoli. As the solubility of CO2 is 20 to 25 times higher than that of O2, the amount of CO2 that can diffuse through the diffuser membrane per unit difference in partial pressure is much higher compared to that of O2. The diffusion membrane is made up of three major layers. Figure 14.4, namely a thin squamous epithelium of alveoli, the endothelium of alveolar capillaries, and the basement substance, composed of thin basement membrane supporting the squamous epithelium and basement membrane surrounding a single pair of endothelial cell capillaries in between them. However, its total thickness is much less than a millimeter. Therefore, all the factors in a body are favorable for diffusion of O2 from alveoli to tissues and that of CO2 from tissues to alveoli. Figure 14.3, diagrammatic representation of exchange of gases at the alveolus and the body tissues with blood and transport of oxygen and carbon dioxide. Inspired air, expired air. Alveolar air, partial pressure of O2, 104 millimeter per millimeter Hg. Partial pressure of CO2, 40 millimeter Hg. In alveolus, CO2 and O2 diffusion, the CO2 goes from the alveolar The CO2 goes from the blood vessels into the alveoli, and the O2 goes from the alveolus to the blood vessels. They travel through the pulmonary vein, systemic arteries carrying oxygenated blood. Partial pressure of O2 95 millimeter Hg, and partial pressure of CO2 40 millimeter Hg. At the level of tissues, the O2 diffuses into the tissues, and the CO2 from the tissue diffuses into the blood vessels. In the systemic veins carrying deoxygenated blood, partial pressure of O2 is 40 millimeter Hg and partial pressure of CO2 is 45 millimeter Hg. Pulmonary artery and then up into the alveolus. Figure 14.4, a diagram of section of an alveolus with a pulmonary capillary. Air, squamous epithelium of alveolar wall, one side first. Blood capillary, red blood cell, endothelium of the blood capillary, basement system, alveolar cavity. 14.4 Transport of gases. Blood is the medium of transport of O2 and CO2. About 90% of the O2 is transported by the RBCs in the blood. The remaining 3% of O2 is carried in a dissolved state to the plasma. Nearly 20 to 25% of CO2 is transported by the RBCs, whereas 70% of it is carried as bicarbonate. About 7% of CO2 is carried in a dissolved state to plasma. 14.4.1 Transport of oxygen. Hemoglobin is a red-colored iron-containing pigment present in the RBCs. O2 can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry a maximum of 4 molecules of O2. Binding of oxygen with a hemoglobin is primarily related to partial pressure of O2, partial pressure of CO2, hydrogen ion concentration, and temperature are the other factors which can interfere with this binding. A sigmoid curve is obtained when the percentage saturation of hemoglobin with O2 is plotted against the partial pressure of O2. This curve is called the oxygen dissociation curve, figure 14.5, and is highly useful in studying the effect of factors like partial pressure of CO2, H plus concentration, etc., and binding of O2 with hemoglobin. In the alveolar, where there is high partial pressure of O2, low partial pressure of CO2, less H plus concentration, and lower temperature, the factors are all favorable for the formation of oxyhemoglobin. Whereas in the tissues with low partial pressure of O2, high partial pressure of CO2, high H plus concentration, and higher temperature exist, the conditions are favorable for the dissociation of oxygen from the oxyhemoglobin. This clearly indicates that oxygen gets bound to hemoglobin in the lung surface and gets dissociated at the tissues. Every 100 ml of oxygenated blood can deliver around 5 ml of O2 to the tissues under normal physiological conditions. Figure 14.5 Oxygen dissociation curve. On the y-axis, we have the percentage saturation of hemoglobin with oxygen. On the x-axis, we have the partial pressure of oxygen in millimeter Hg. We obtain a sigmoid curve. 14.4.2 Transport of carbon dioxide. Carbon dioxide is carried by the hemoglobin as carbaminohemoglobin, about 20 to 25%. This binding is related to the partial pressure of CO2. Partial pressure of O2 is a major factor which could affect the binding. When partial pressure of CO2 is high and partial pressure of O2 is low, as in the tissues, more binding of carbon dioxide occurs, whereas the partial pressure of CO2 is low and partial pressure of O2 is high, as in the alveolar, dissociation of CO2 from the carbamino hemoglobin takes place. That is, CO2, which is bound to hemoglobin from the tissues, is released at the alveoli. RBC contain a very high concentration of the enzyme carbonic anhydrase and a minute quantities of the same is present in the plasma too. This enzyme facilitates the following reactions in both directions. CO2 plus H2 in presence of carbonic anhydrase forms carbonic acid. Again, in presence of carbonic anhydrase, they split into bicarbonate ion plus H plus ion. At the tissue site, where the partial pressure of CO2 is high due to the catabolism, CO2 diffuses into blood, RBCs and plasma and forms the bicarbonate and H plus ion. At the alveolar site, where the partial pressure of CO2 is low, the reaction proceeds in the opposite direction, leading to the formation of CO2 and water. Thus, CO2 trapped as a bicarbonate at the tissue level and transported to the alveoli is released out as CO2. Figure 14.4. Every 100 ml of the oxygenated blood delivers approximately 4 ml of CO2 to the alveoli. 14.5 Regulation of respiration. Human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissues. This is done by the neural system. A specialized center present in the medulla region of the brain, called respiratory rhythm center, is primarily responsible for this regulation. Another center present in the pons region of the brain, called pneumotactic center, can moderate the functions of the respiratory rhythm center. Neural signal from this center can reduce the duration of inspiration and thereby alter the respiratory rate. A chemosensitive area is situated adjacent to the rhythm center, which is highly sensitive to CO2 and hydrogen ions. Increase in these substances can activate the center, which in turn can signal the rhythm center to make necessary adjustments in the respiratory process, by which these substances can be eliminated. Receptors associated with the aortic arch and carotid artery also can recognize changes in CO2 and H plus concentrations and send necessary signals to the rhythm center for remedial action. The role of oxygen in the regulation of respiratory system is quite insignificant. 14.6 Disorders of the respiratory system. Asthma is a difficulty in breathing causing wheezing due to inflammation of bronchi and bronchioles. Emphysema is a chronic disorder in which alveolar walls are damaged, due to which respiratory surface is decreased. One of the major causes of this is cigarette smoking. Occupational respiratory disorders. In certain industries, especially those involving grinding or stone breaking, so much dust is produced that the defense mechanism of the body cannot fully cope with the situation. Long exposure can give rise to inflammation, leading to fibrosis. Proliferation of fibrous tissue can thus cause serious lung damage. Workers in such industries should wear protective masks. Summary: cells utilize oxygen from metabolism and produce energy along with substances like carbon dioxide, which is harmful. Animals have evolved different mechanisms for the transport of oxygen to the cells and for the removal of carbon dioxide from them. We have a well-developed respiratory system comprising two lungs, associated air passages, and to perform this function. The first step in respiration is breathing, by which atmospheric air is taken in, inspiration, and the alveolar air is released out, expiration. Exchange of O2 and CO2 between the deoxygenated blood and alveoli. Transport of these gases throughout the body, alveoli. Exchange of O2 and CO2 between the oxygenated blood and tissues and utilization of O2 by the cells. Cellular respiration are the other steps involved. Inspiration and expiration are carried out by creating pressure gradients between the atmosphere and the alveoli with the help of specialized muscles. Intercostals and diaphragm. Volume of air involved in these activities can be estimated with the help of spirometer and are of clinical significance. Exchange of O2 and CO2 at the alveolar and tissues occur by diffusion. Rate of diffusion is dependent on the partial pressure gradients of O2, partial pressure of CO2, their solubility as well as the thickness of the diffusion surface. These factors in our body facilitate diffusion of O2 from the alveoli to the deoxygenated blood, as well as from the oxygenated blood to the tissues. The factors are favorable for the diffusion of CO2 in the opposite direction, that is, from the tissues to alveoli. Oxygen is transported mainly as oxyhemoglobin. In the alveoli, where the partial pressure of O2 is higher, O2 gets down to hemoglobin, which is easily dissociated at the tissues. The partial pressure of O2 is low and partial pressure of CO2 and excess concentration are high. Nearly 70% of the carbon dioxide is transported as bicarbonate with the help of enzyme carbonic anhydrase. 20 to 25% of the carbon dioxide is carried by hemoglobin as carbaminohemoglobin. In the tissues where the partial pressure of CO2 is high, it gets down to blood, whereas the alveoli, where partial pressure of CO2 is low and partial pressure of O2 is high, it gets removed from the blood. Respiratory rhythm is maintained by respiratory center in the medulla region of brain, a pneumotaxic center in the pons region of the brain, and a chemosensitive area in the medulla can alter respiratory mechanisms. Thank you, and stay tuned for the next chapter, which will be body fluids and circulation.